All right, so the question says, which of the following most closely describes a Hoogsteen position? Okay, and feel free to go over the answer choices here, and we're gonna go over them one by one. The option number A says the interaction of four guanine bases from different strands. Well, um, this describes a G quadruplex, right? So let me write it down. So it describes a G quadruplex. And what it is, it's also called a G tetrad. And it's a specific type of four-stranded DNA structure, right? So this one certainly does utilize Hoogsteen type hydrogen bonding to hold four guanine together in a square plane, right? But the description itself defines a quadruplex, and it's not like a general definition of Hoogsteen position or interaction. So I would eliminate this option. And then number B says a nucleotide pair forming an H-bound interaction with an additional nucleotide from a third strand. Well, this option looks correct, and here's why. So, first of all, we want to know what exactly is a Hoogsteen position or a Hoogsteen phase, and I'm gonna circle it up. So generally, when you base pair, um, you have, for instance, you have guanine over here, and you're base pairing it with cytosine, right? When you base pair, um, this guanine over here, its major phase is actually those three, and those three are gonna be the main component that is gonna form the hydrogen bound, and it's gonna. So this one is actually going to base pair with this one, and then this one's gonna base pair with this one, and this one's gonna base pair with that one, right? And that is standard, but in Hoogsteen, the Hoogsteen phase involves something that's over here. So that involves hydrogen bonding utilizing this non-major phase of guanine. And this is the same for adenine too, which also utilize this nitrogen over here, and that's nitrogen number 7. And I will circle the whole thing, right? So this will be a Hoogsteen phase of the adenine, and this will be a Hoogsteen phase of guanine, right? So this one will be Hoogsteen phase of guanine. So those are going to be the Hoogsteen phase, and those phases are where some additional nucleotide from its third strand can actually slide into the major groove and form hydrogen bond with those Hoogsteen phase of an existing Watson-Crick base pair. So you can have guanine base pairing with cytosine, but then the Hoogsteen phase over here, this is still going to be exposed. The Hoogsteen phase is still going to be exposed, and it's going to be able to form an additional hydrogen bond with a third nucleotide, forming something called a triple X DNA. So this one is the correct option. Let's look at number C, which says a primary sequence with a high probability of forming a hairpin. Well, this hairpin loop actually forms from standard Watson-Crick base pairing. So this one is standard Watson-Crick base pairing, and that's when a single strand of DNA or RNA folds back on itself due to a palindromic sequence. So that's not correct. And then option number D says a protonated cytosine. While a protonated cytosine is often like you have this one protonated here, right? This cytosine, you have this protonated here. And this is often a requirement to form a Hoogsteen pair with a guanine in parallel triplexes. For instance, if you want to utilize this phase of guanine over here, right? If you want to utilize that phase, let me use a different color. So you want to utilize this phase over here. You want to pronate this so that these two can hydrogen bond with each other, right? I guess yellow isn't that clear, so I'm actually going to use red. So these two are gonna hydrogen bound, are gonna be able to hydrogen bound, and these two can now hydrogen bound with each other like that. So that is utilizing the Hoogsteen position, and you want a protonated cytosine, but it doesn't really describe a Hoogsteen position. What a Hoogsteen position is not a protonated cytosine, but rather those phases that's exposed, right? Even when that's not involved in standard base pairing. So we can eliminate option number D. And then E says a secondary structural element associated with gene regulation. Well, Hoogsteen pairings can play a role in regulation, right? This option doesn't really describe a Hoogsteen position. It describes a biological function, but not the chemical signature or chemical structure of a Hoogsteen position. So we can also eliminate this one. So the final answer is that a Hoogsteen position, it's a nucleotide pair forming an H-bond interaction with an additional nucleotide from a third strand. All right, so that's it for this question. Thanks for watching.